this video, I'll be using Minecraft to explain Methanogenesis by Archaea. Organisms that perform methanogenesis are referred to as methanogens and are also extremophiles, so they prefer to live in extreme environments such as really hot or cold environments, acidic environments. They also thrive in anaerobic environments, and all of this is due to their rigid cell wall. One of the environments that you can find them in is a submarine volcano, which, as you see here, these purple blocks in Minecraft, if you put lava underwater, it forms that purple block. It's called obsidian, so that kind of just shows that that's a submarine volcano. Another environment that they thrive in would be wetlands, which is why I built my diagram in the swamp biome. They're also one of the main causes of marsh gas production, which is a greenhouse gas. They also live in the gut of animals. The methane gas that they produce forms into flatulence. So here I have a cross-section of an Archaea cell, and within that I have the methanogenesis pathway all drawn out here. So starting from the top, we've got carbon dioxide gas, and this will get reduced by ferredoxin R, which means the reduced form of ferredoxin, which comes from this pool down here that actually is created using a cell membrane pump. Both of these reducing cofactors are actually products of different pathways. So up here, the reduced ferredoxin comes in, reduces carbon dioxide, and forms oxidized ferredoxin. And in the same step, the CO2 is ligated to methanofuran. And after both of these occur, we're left with Formyl-methanofuran. This is then moved, the Formyl group is actually moved off of the methanofuran by a transformylase, and it's actually moved to a tetrahydromethanopterin group, which, as you can see here, there's that group coming in and this group leaving, which leaves us with a Formyl-tetrahydromethanopterin. The next step, this molecule gets cyclized by a cyclohydrolase, and in this step we lose a water molecule. And this leaves us with Methenyl-tetrahydromethanopterin. And then once we have that, there's two different steps we can go through to reduce it. We can either have an enzyme just add electrons to it to reduce it, or we can use the F420H2 from this pool here to reduce it, forming the F420, which is the oxidized form, resulting in a Methylene-tetrahydromethanopterin. This then gets reduced by another F420H2, forming a Methylene-tetrahydromethanopterin. Methyl-tertrahydromethanopterin. Then this methyl group gets moved to a HS-CoM by an S-methyltransferase. This step occurs in the cell membrane with the use of a sodium pump. Then an HS-CoB comes in and binds to the HS-CoM, basically causing the methyl group to be a good leaving group and kicking it off completely, which then results in the form of to form methane. So that concludes this pathway. I will put in the description box what all of these abbreviations mean, just for future reference. Thank you all for watching.